Let's watch an experiment performed on a hydrilla plant. The hydrilla plant is kept in a beaker filled with water. A funnel is placed over it. A test tube is inverted over the stem of the funnel. Let's keep this setup in two different conditions, in light and in dark. When the setup is kept in light, we observe that many bubbles evolve continuously into the inverted test tube. These bubbles are actually oxygen that is released during photosynthesis in hydrilla. When the setup is kept in dark, the number of bubbles will be comparatively less. Why is this so? Why is there a difference in the number of oxygen bubbles when the plant is kept under different conditions of light? Light is important for the light-dependent reaction of photosynthesis, during which oxygen is released. In the absence of light, the hydrilla's rate of photosynthesis was less, and hence there was a decrease in the number of oxygen bubbles. So, from this experiment, we can conclude that light intensity affects photosynthesis. But how do you confirm that the bubbles released are oxygen bubbles? There's a solution to it. After bubbles collect in the inverted test tube, carefully remove the tube and place a glowing splinter in the test tube. The splinter will light up with a flame. Oxygen in the tube helps ignite this flame. This proves that oxygen was produced as a result of photosynthesis of hydrilla. There are many factors apart from light that affect photosynthesis. These factors are classified as external and internal factors. External factors are the environmental factors that affect photosynthesis. It includes light, carbon dioxide, temperature, water. Internal factors include leaf anatomy, chlorophyll content, age of the leaf, and accumulation of photosynthetic products. All these factors affect the rate of photosynthesis. If any one factor is deficient, photosynthesis rate will be affected. The factor that is deficient or is present only in suboptimal levels becomes the limiting factor and will be responsible for affecting the rate of photosynthesis. The rate of photosynthesis is generally calculated based on the rate of oxygen production. However, this is an approximate estimation, as some of the oxygen produced in the process of photosynthesis is used by the plant for respiration. Starch production is an indicator of the occurrence of photosynthesis. Let's check out how to determine the presence of starch in a leaf. Destarch the leaves of a potted plant by keeping the plant in the dark for 2 to 3 days. Pluck a leaf off it and boil it in water. Next, pour off the water and boil the leaf in ethanol or methanol over a boiling water bath. Keep boiling until the leaf becomes pale white. This indicates that chlorophyll is removed. This procedure causes the leaf to become brittle. Soften the leaf by dipping it in warm water. Now, place the leaf on a plain surface and pour a few drops of iodine solution on it. Carry out the same procedure on a leaf plucked from a plant kept in sunlight. Then subject it to iodine solution. You will observe that the leaf from the plant kept in sunlight turns bluish black in color, while the other leaf wouldn't show any color change. This iodine test indicates that the leaf from the plant kept in sunlight was actively photosynthesizing and producing starch, whereas the plant, when kept in dark, couldn't photosynthesize and hence starch was not produced in its leaves. Let's learn more about some of the external factors that affect the rate of photosynthesis. Let's begin with light. The rate of photosynthesis is affected by the intensity of light. Photosynthesis occurs in the visible range of the light spectrum between 400 to 700 nanometers. Maximum photosynthesis occurs in blue and red light. Rate of photosynthesis will increase if the light intensity is increased, but it won't increase beyond a certain point, because according to Blackman's law of limiting factors, when a process depends on a number of factors, its rate is limited by the pace of the slowest factor. Let's look at an experiment to see how deficiency of light can affect photosynthesis. Take a plant with de-starched leaves. Cover a part of a leaf with black paper and leave the plant in sunlight for a few hours. Then pluck the covered leaf and test it for the presence of starch. You will observe that the part which was covered with black paper will not show any color change, whereas the other parts of the leaf which were exposed to sunlight will show a positive starch test. What can you conclude from the experiment that you just saw? The part which was covered with black paper failed to receive light and hence couldn't photosynthesize. The experiment thus shows that light can be a limiting factor that affects photosynthesis. Let's move on to the next factor, carbon dioxide. Rate of photosynthesis will increase if the carbon dioxide concentration is increased. But again, if the plant is not receiving the other essential requirements in a proportionate rate, photosynthesis rate won't increase. Blackman's law of limiting factors is applicable here too. Let's look at an experiment to see how deficiency of carbon dioxide can affect photosynthesis. Take a plant with de-starched leaves. Take a conical flask containing some potassium hydroxide, KOH solution. Insert a de-starched leaf into this conical flask through a split cork on the mouth of the flask. Leave this set up in light for a few hours. Then remove the leaf from the flask and test it for the presence of starch. What will you observe? Remember that potassium hydroxide absorbs all carbon dioxide from the flask. As there was no carbon dioxide left in the flask, the leaf wasn't able to photosynthesize and starch wasn't synthesized. As a result, the leaf will show a negative starch test. This proves that even carbon dioxide concentration can affect photosynthesis. Another external factor affecting photosynthesis is temperature. Enzymatic reactions are affected by changes in temperature. Enzymes require an optimum temperature for their functioning, beyond which they will denature. Optimum temperature for photosynthesis lies in the range of 20 to 35 degrees centigrade. The next important factor to consider is the availability of water. Water is absorbed through the roots, and excess water can cause the roots to rot, while insufficient water will hamper photosynthesis. If water is insufficient, the stomata will close to prevent loss of water. This will affect the intake of carbon dioxide and hence photosynthesis will be affected. Let's learn about some of the internal factors that affect photosynthesis. The first being the leaf structure. As photosynthesis occurs in the leaves and green parts of the plant, most of the internal factors are concerning the leaves. The leaf structure influences photosynthesis because, after all, the leaf is the major organ for photosynthesis. The number and distribution of stomata, the thickness of the cuticle, and the surface area of the leaf influence the rate of photosynthesis because these are responsible for the exchange of gases and the transpiration of water. Chlorophyll, the light-trapping molecule of photosynthesis, is important. The amount of chlorophyll will affect the rate of photosynthesis. Have you ever wondered how variegated leaves, such as that of coleus, carry out photosynthesis? The green parts of variegated leaves contain some amount of chlorophyll, and these parts carry out photosynthesis. So, let's perform an experiment with a variegated leaf and prove that it won't be able to photosynthesize if it didn't have chlorophyll. Pluck a leaf off a coleus plant. Mark the green and non-green areas of the leaf and then perform the starch test on it. You will observe that the non-green areas of the leaf test negative for starch, whereas the green areas show bluish-black color. This indicates that the green part of variegated leaves carried out photosynthesis. Photosynthesis was possible only because of the presence of chlorophyll in the green parts. The age of the leaf also affects the rate of photosynthesis. The rate increases as the leaf matures. It's maximum in a mature leaf, which gets all other requirements in optimum amounts. If starch gets accumulated in plant cells, it's an indicator to slow down photosynthesis. So, the accumulation of photosynthetic products also affects photosynthesis. Thus, all these external and internal factors act as a team and perform photosynthesis.